Here we go. Let me just briefly introduce you. Welcome here, Jamal. It's a pleasure to have you. And Elicera Therapeutics, and you are the CEO. It is a pleasure for us to have you. It will be in English, and please ask questions in the lower chat field. It will be a 10 minutes introduction and then some questions, Jamal. So I hope you're ready.
I'm ready. Thank you. Thank you for the invitation to hold a presentation on Elicera. I will try to keep it within 10 minutes. So my name is Jamal Mosle. I'm the CEO of Elicera Therapeutics, which is a cell and gene therapy company focusing on immuno-oncology. This means that we develop different types of therapies that uses the patient's own immune system to fight cancer. We develop two types of therapies, oncolytic viruses and CAR T cells. And we also have a fully developed technology platform, which we call iTANK, that can be used to further enhance the efficacy of these therapies. So what the iTANK platform does basically is that it's a genetic modification of these therapies that makes it possible for them to elicit a parallel immune response and activation of the patient's own killer T cells in parallel to the cancer attack by oncolytic viruses and CAR T cells. We have experience in the board and management, both from immuno-oncology, cell therapies, business development, and also important areas such as regulatory within biologics and cell and gene therapy, and also CMC, which is production of these types of complex treatments. So that's important for a company such as Elicera. On the next slide here, you see a picture of our pipeline. You see that we have 4 different projects under different development stages. The most advanced is what we call ELC100. It's an oncolytic virus that we currently are testing in a clinical study in neuroendocrine tumors. So we're basically halfway past that study. We've treated 7 patients out of 12 planned in this step here. We haven't seen any adverse events, and we've actually already reported promising efficacy data from one of the patients. ELC201 is our next generation oncolytic virus, which we have genetically modified so that it elicits a combination of 3 modes of actions in one product. It also is a product that can be applied for treatment of most cancers. 301 and 401 are our CAR T cells under development. 301 is in liquid tumors. Initially, we will target B-cell lymphoma in a clinical study that we plan to initiate next year. And 401 is initially in glioblastoma multiforme, which is an aggressive form of brain cancer. But this product we also believe has the potential to treat an additional other solid tumors such as melanoma, head and neck cancer, pancreatic cancer, and also colon cancer. The iTANK platform is included in 201, 301, and 401. And it's also a technology platform which is actually applicable to any CAR T cell under development. So our business model is based on out-licensing both our technology platform and our drug candidates. And the strategy for out-licensing the drug candidates is to build value and minimize risk by completing at least clinical phase 1/2 studies on our own, and then seek to potentially partner these candidates. The iTANK platform, however, being that it's fully developed and that it solves 2 of the major problems that CAR T developers face in the treatment of solid tumors, we believe that this technology is highly interesting for basically everybody that develops CAR T cells. So it's something that we intend to, or actually have already started the process of looking for licensing and partners for this specific platform. For the other ones, we have products that are both more niched and more broad. I've mentioned it before, but quickly here again. So 301 and 100 are both a little bit more niched in a few cancer indications, while 401 and 201 have a potential across multiple cancer indications, giving us multiple opportunities for out-licensing. So what is an oncolytic virus? It's a virus that you have genetically modified so that it selectively infiltrates and replicates only in cancer cells, but not in healthy cells. So in this way, you get a oncolysis, which is a tumor cell death due to the fact that the tumor cell bursts when you have enough replication of the virus inside the tumor cell. This is a process that in itself starts a parallel immune response by activation of the patient's own T cells, which is the patient's, you can say, immune system soldiers. So you get a two-way attack on cancer this way, both by the virus particles continuing to infiltrate other cancer cells, and then you have the parallel attack by the T cells. The difference between our two oncolytic viruses is that ELC-100 is restricted to neuroendocrine tumors and elicits two mode of actions, the T cells and the virus attack, while 201 is applicable for treatment against most cancer indications, and we have genetically modified it so that it elicits a three mode of action in one product. The study that is ongoing, as I mentioned, is in 12 patients. It's a so-called dose escalation study where we intend to look for maximum tolerable dose in these 12 patients to then be tested in an additional 12 patients that will get the maximum tolerable dose. So it will be done in two steps here. So no reported adverse events, and we've already seen that one of the patients has reported partial response, which is encouraging since this patient belongs to a dose group which is quite low. So you can expect the efficacy to increase as you increase dose. But this is to be seen. If we go in and talk a little bit about CAR T cells, we can mention that immuno-oncology is quite a broad term. So the term includes many different ways of using the patient's immune system to fight cancer. And CAR T cell therapy is among one of the most interesting in this field of immuno-oncology. In fact, ASCO, which is one of the world's largest cancer organizations, they named CAR T cell therapy as Advents of the year 2018. And the reason they did this is because it was shown that CAR T cell therapy was able to actually cure the majority of patients that were previously not eligible for any other treatment, that have undergone even 2 lines of treatment before and didn't respond to them. These patients, a lot of them, you could eradicate all tumors. So this has worked very well. And the way this functions is that you take out blood from the patient, you filter out T cells from the patient, and then you genetically modify them so that they express a chimeric antigen receptor, a CAR molecule on the surface. And this CAR molecule then recognizes certain targets on the tumor cell. You expand them in test tube and then you re-inject them into the patients. And the idea is that they will then find the tumor cells that carry this target and attack them. It's worked very well in liquid tumors, but unfortunately it hasn't worked so well in solid tumors. And there's 2 major reasons for that. A solid tumor has a very hostile tumor microenvironment. And this hostile microenvironment, it inhibits the effect of the CAR T cell and lowers its function. So it doesn't have enough efficacy in solid tumors. The second major reason is that it's difficult to find a target that is expressed on all tumor cells in a solid situation. This means that if the CAR T cell only targets a target that is expressed on some but not all tumor cells, the tumor cells that lack the target, they will of course escape and will not be attacked by the CAR T cell, and they will form CAR T resistant tumors. So this is a big issue. And the iTANK platform, we can actually solve both of these issues here. So with the iTANK platform, we elicit a immune response that directly counteracts the hostile tumor microenvironment and strengthens the function of the CAR T cell. And we can generate a activation of the patient's own killer T cells against a whole repertoire of different targets on the tumor cell. There's of course a very large interest in the CAR T cell field in this industry. There's over 100 companies developing CAR T cells worldwide. It's a rough 50-50 split between China and US. You have a little bit more companies in China developing CAR T cells. Not that many in Europe, only 14, and only 1 in Sweden, which is Elicera. The 5 approved CAR T cells today, market approved, they are only approved for treatment of liquid tumors. So far, none of these therapies have been approved in solid tumors. due to the two reasons that I mentioned before. Most of the CAR T cells are still in early development, so a company such as Elicera still has a lot to do in this field. To further put this into perspective, we can mention that CAR T cells have been developed in different generations. First generation were shown to have an insufficient proliferation, a brief T cell survival, and an insufficient immune activation. And due to this, we continue to make these CAR T cells better in different generations and up until fourth generation. What differs a fourth generation from other generation CAR T cells is the fourth generation contains a gene that codes for an immune stimulating factor. Usually it's a cytokine 1, and usually it's IL-12. So that triggers the immune response further. And Elicera develops fourth generation CAR T cells due to our iTANK platform. And what that means and what differs our from other fourth generation is that instead of including a gene that codes for a cytokine, we include a gene that codes for NAP, which is short for neutrophil activating protein from Helicobacter pylori. So when NAP is activated, it actually releases over 20 different cytokines and chemokines, which we believe gives a much broader and effective immune response against cancer. And it's this big release of cytokines and chemokines that inhibits the immunosuppressive tumor microenvironment and instead creates a very pro-inflammatory, good environment in the tumor for the immune system. Most of the CAR T cells under development today are still second generation, as much as 70%. And only 16% are fourth generation. And of these 16%, most of them are not companies. They're actually developed in academia. So Elicera as a company, we believe that we are one of two that develops this in the world. We've only identified one other company in Japan that develops a fourth generation CAR T cell. So this slide only shows you the benefit that I've already described shortly. So normally when you have a CAR T cell, it's the CAR T cell that attacks the cancer, and this is how you kill the cancer cells. So you're on number one here. But with the iTANK platform, we also, of course, have the CAR T cell killing cancer cells, but we also then, in number two here, generate a second mode of action through activation of the patient's own killer T cells, not only against the target that the CAR T cell is directed against, but the whole set of different targets on the tumor cell. We've tested this quite extensively in different preclinical studies showing proof of concept for the iTANK platform. They have recently been presented at two scientific conferences, ESGCT and the PEGS conference here in October and November this year. So we have tested this in animal studies, and we have compared the results with the treatment with conventional CAR T cells, meaning that they are not armed with the iTANK platform. And we have done this in different tumor types, in different mouse models, and also with different CAR molecules. And regardless, we see an increase in survival and a decrease in tumor growth when you treat them with iTANK enforced CAR T cells in comparison to conventional. So this shows you that this is a platform that is universally compatible with other CAR T therapies. So this is why we say that it's important for other companies to also use this technology platform to strengthen their products. So to finish here, this is a slide of our clinical development plan for the coming 3 years. You see that we have an ongoing clinical study that will be completed the first step next year. We also intend to have additional studies being initiated during 2023, so we'll have quite an extensive clinical program by then. Our next clinical study to be initiated is in B-cell lymphoma with the 301, which is our CAR T cell in liquid tumors. We listed the company, I should say, also on Nasdaq First North Growth Market here in Sweden this year in June. So we have enough funding to last at least until mid-2023 and across some important milestones.
Jamal, perhaps, yeah, please. That I think that's we have approximately 2 minutes left, so that's a good way of finishing off. Thank you.
Okay, should I take the 2 minutes to?
Yes, please. The investment case, because that's what people are asking for.
Good. Okay, so very short. To summarize everything, we have a broad project portfolio with a diversified pipeline of technologies. So we have both CAR T cells and oncolytic viruses. We have a next generation oncolytic virus and what we call an optimized version of 4th generation CAR T cells. So we're very much in the forefront of cancer research in cell and gene therapy. We have a fully developed iTANK platform for treatment of cancer using immune enhancement of oncolytic viruses and CAR T cells. It is fulfilling a large unmet medical need in several cancer forms. So a lot of our products can be used across multiple indications, meaning that we also have a flexible and attractive business model in the fact that we can also have one product, but multiple licensing for that specific product. I haven't talked so much about our IP portfolio, but of course we have patent applications that protects the iTANK technology and our drug candidates. And we have an experienced management and board of directors. And I can mention myself, I've previously been CEO and co-founder of another immuno-oncology company for almost 10 years. It's listed on a small cap in Sweden called Immunicum. And we also have our CSO and co-founder, Magnus Sand. He has previous clinical experience from both CAR T cells and oncolytic viruses, which is very important, of course. So I will end with that and will answer any questions you might have.
Yeah, we're running a bit tight here, Jamal, so I'm sorry for that. But I think it's a fair question that there's many companies in this field, not in this field, but in early stage pharma or biotech. So if you want to invest in your company, of course, you are putting a lot of things here, but can you mention any other reasons that you think that you should be looking at your company compared to others perhaps?
Yeah, I appreciate the question. So the main thing that I would like to point to what differentiates us from other companies, especially when looking into the CAR T field here, is the fact that we have the iTANK platform. So as I said, there are other CAR T developers, but all of them, I should say most of them actually focus on liquid tumors because that's the easy area. The biggest potential is in solid tumors.
Yeah.
And in solid tumors, everybody's facing the same problems with the immunosuppressive tumor microenvironment and the problem with antigen heterogeneity, meaning that there's a lack of good targets in solid tumors. And the iTANK platform is very unique in the sense that it can meet both of these challenges. And there's not a lot of companies that can claim what we are claiming with the iTANK platform. So we use the iTANK platform for our own sake, but we can also, as I said, we can offer that technology by non-exclusive licenses to other companies that develop CAR T cells. So in this way, we believe that we can potentially generate income for the company in the short term, and we can do that multiple times. Yeah.
Jamal, thank you very much for your presentation. I'm sorry the time is up. So it was meant to be a brief introduction. So you've done so. Thank you very much. Hope to see you some other time and have a good day.
Thank you.
Thank you.